Welcome to EQBloom. Today we journey into one of the most iconic structures in all of biology, the nucleus, often called the control center of the cell. But what does that really mean? The nucleus isn't just a static bubble of DNA, it's a dynamic, decision-making hub. Every heartbeat, thought, or immune response begins with instructions encoded in this glowing, coiled vault. In this video, we'll explore how DNA is packaged, how genes are regulated, how proteins are made, and why the nucleus is not only for life, but for identity, development, and disease resistance. The nucleus is exclusive to eukaryotic cells, the more complex cells that make up everything from fungi to humans. It is surrounded by a double membrane called the nuclear envelope, which safeguards the DNA while still allowing controlled exchange with the rest of the cell. Inside, you'll find chromatin, a mix of DNA and proteins, and a prominent structure called the nucleolus, where ribosomes are assembled. The nuclear envelope contains pores, tiny gateways that regulate molecular traffic. Interestingly, not all cells have a nucleus. For example, mature red blood cells expel their nucleus to make room for more hemoglobin, enhancing their oxygen-carrying capacity. But for most cells, the nucleus is indispensable. It's where the blueprints for everything the cell does are kept, accessed, and updated. Let's talk scale. Each human cell contains around 2 meters of DNA. That's a miraculous feat of compression and organization. DNA wraps around proteins called histones, forming structures that resemble beads on a string. These beads, nucleosomes, coil further into chromatin fibers, which then fold into the familiar X-shaped chromosomes during cell division. But DNA isn't uniformly packed. Loosely packed regions called euchromatin are accessible and actively transcribed. Tightly packed areas, heterochromatin, are silenced or inactive. This isn't random. It's a key part of gene regulation, controlling which genes are turned on and when. The organization is so precise that it can change dynamically, loosening to express a gene when needed, then tightening again for storage or protection. Packaging is not spatial efficiency, it's biological intelligence. Every cell in your body has the same genome, yet a neuron behaves nothing like a skin cell. How? Gene regulation. This system controls which genes are expressed, meaning which are transcribed into RNA and which are kept silent. At the heart of this system are transcription factors, proteins that bind to specific DNA sequences near a gene and decide whether it's activated or repressed. Think of it like a set of light switches. Only some are flipped on at a given moment, depending on the cell type, environment, or stage of development. This is also where epigenetics enters the picture. Chemical tags like methyl groups, acetyl groups can fine-tune gene activity without altering the DNA code itself. This mechanism is how identical twins can develop differently, and how your environment, stress, diet, toxins can literally reprogram gene expression. The nucleus is not just for storing instructions, it's constantly deciding what parts of your genetic library to read. The central dogma of molecular biology begins here, DNA to RNA to protein. The first step, transcription, takes place inside the nucleus. When a gene is turned on, an enzyme called RNA polymerase binds to the DNA and unzips a portion of the helix. It then copies one strand of the DNA into a molecule of messenger RNA, mRNA. But this isn't a carbon copy. An RNA, thymine, is replaced with uracil, so base pairing looks like AU and GC. Once the mRNA is built, it exits the nucleus through a nuclear pore and travels to the cytoplasm, where the next step, translation into protein, takes place. Transcription is the nucleus's way of exporting instructions. Every protein your body makes starts with this step. The nuclear envelope might look like a solid wall, but it's actually perforated with complex gateways called nuclear pores. These act as highly selective filters. Large molecules like mRNA can only exit when properly tagged. Meanwhile, proteins like enzymes, transcription factors, or stress signals are actively imported into the nucleus when needed. This controlled flow maintains the security of your genetic material while enabling constant communication with the rest of the cell. Errors in this transport system can be catastrophic. Certain cancers, autoimmune disorders, and viral infections exploit or corrupt nuclear transport. For instance, viruses like HIV insert their own genetic material into the nucleus to hijack the host machinery. So these pores aren't just passive holes. They are intelligent gatekeepers in your cell's most secure vault. Let's zoom out. The nucleus isn't just a container. It's the conductor of a cellular symphony. It reads the score, assigns instruments, modulates intensity, and edits in real time. It is where the script of life is archived, read, edited, and sometimes rewritten. What makes you you, your height, immune traits, even your temperament, begin as chemical instructions activated inside this tiny organelle. And yet this power must be wielded with incredible precision. Misfiring genes can cause cancer. Faulty regulation can lead to autoimmune diseases, but when it all works, when genes turn on at just the right moment, the result is life, functioning, adapting, thriving. So the next time you think about who you are, zoom in to your cells and within them to the nucleus, where your past is stored, your present is managed, and your biological future is quietly orchestrated.